Hi class, in this recording we're going to focus on neurons, the cell, the most important single unit of the nervous system. We could think of the neuron as the structural and functional unit of our body's nervous system. Now as we look at a neuron, there's some unique properties of all neurons. For instance, when we think of our neurons are excitable or irritable. What does that mean? That means they can respond to changes in the environment. These changes in the environment are referred to as stimuli in our environment. They can be temperature changes, pressure changes, chemical gradient changes. There's lots of different things that can excite a neuron. And once it's excited, it initiates this action potential. So when we think of this excitability, I want you to think of it as initiating electrical charges. And this is something that's very unique to just neurons. Only neurons can initiate electrical charge production or action potentials, and that's unique to the neuron. Other muscle or other cell types in the body, like muscles, for instance, can conduct electricity, but they cannot initiate the signal that's being conducted. So when we think of neurons, not only can neurons initiate the signal, or not only are neurons excitable, neurons, once they initiate the signal, they can conduct that electrical signal. They can respond to the stimuli by producing the signals that are then conducted to other cells. And then we have secretion. Once that signal reaches the end of the neuron, the axon terminal, the axon is also known as a nerve fiber. A neuron is not a nerve. A bunch of axons from neurons will make a nerve. So when the signal reaches the end of the nerve fiber or the end of the axon of the neuron, then there's going to be some chemicals released. Those chemicals are referred to as neurotransmitters, and they are going to influence the next cell downstream, either to produce an action potential potentially or not produce an action potential, or maybe carry out some kind of a secretion. So as we look at our neurons, we can categorize them based on functional classes. Sensory neurons are going to carry signals to the central nervous system. They access the central nervous system, and they're part of the afferent branch of the sensory division within the peripheral nervous system. They are going to detect stimuli and then transmit that to the brain and spinal cord, the central nervous system. So here we have in our fingertip some sensory neuron. It gets stimulated, the signal travels to the brain and spinal cord, and within the brain and spinal cord, within our central nervous system, we have interneurons. Interneurons are going to be entirely within the CNS, and you can think of them as the processors. Most of our neurons, about 90% of the neurons in our bodies, are interneurons, and they are going to make the decisions. These are the decision makers in our bodies. They carry out the integrative functions. When I say integrative functions, they're just going to receive all the different pieces of information and make a decision based on all those different pieces of information. And as they make decisions, it's going to be with electrical gradients or electrical charges. We can have excitatory charges or inhibitory signals. Excitatory signals or inhibitory signals sent to those neurons. We also have the motor neuron. So after our interneuron makes the decision, it's then going to stimulate a motor neuron, which originates in the central nervous system and then transmits a signal, an action potential or electrical signal, down its axon to a muscle or gland. These are referred to as the effectors, and they are part of the efferent nervous system. The efferent nervous system is going to carry signals away from or exiting the central nervous system, and motor or efferent neurons will be the structural and functional unit of that branch of the nervous system. We also can categorize neurons based on their structure, but before we can talk about the structure of neurons, we need to focus on, or before we can talk about categorizing them based on structure, we need to explain what the structure is. So first we have the neurosoma. The neurosoma can also be thought of as the cell body of the neuron. Sometimes it's called just the soma. Sometimes it's just called cell body of neuron. I tend to be a pretty big fan of the term cell body of neuron, but neurosoma is the full technical name. And within this neurosoma, we're going to have a big fat nucleus. And this nucleus, relative to the size of the neurosoma, is usually fairly large. It's going to take up, or it's going to be about 1/2 the diameter of the neurosoma. And as we look at our diagram, we have a nucleus right here that I'm now outlining in green. So we have the neurosoma. Within the neurosoma, we have the nucleus. And within the nucleus, we have a nucleolus. That nucleolus is going to be fairly large because the neurosoma, or the cell body of the neuron, is going to be making lots of proteins. And to make lots of proteins, it needs lots of ribosomes. And to make lots of ribosomes, it needs a very well-developed nucleolus. The nucleolus is where we make ribosomes within the cells. As we're looking at the neuron, the cytoplasm of the neuron, sometimes referred to as the neuroplasm, I'm not a big fan of that term, it's going to have a lot of mitochondria. Where neurons are very electrically active, and that means they need lots of chemical energy, a lot of lysosomes. The Golgi complex is going to be fairly extensive, and there's lots and lots of rough ER. As we're looking at rough ER, that rough ER is so extensive within the neurosoma, it's visible as dark patches. So if you probably have to squint your eyes and zoom in close if you're looking at a cell phone screen right now. You can see darker regions within the cytoplasm. Those darker regions represent a lot of rough ER. And it's very dark because of the fact that it's going to have a high concentration of ribosomes synthesizing many different proteins that are going to be associated with neurotransmitters. We also have a lot of inclusions within the cytoplasm of the nucleus. These inclusions, for those of you who don't remember, think all the way back to chapter 3 of this class, the second week of class. An inclusion is just kind of like a storage vesicle within the cell. It's going to store different chemical compounds. We can store glycogen. Neurons need lots of carbohydrates. That is their preferred fuel source. We could store lipids. We can store melanin, the pigment. We also can store lipofuscin pigment. This lipofuscin pigment is a byproduct of when lysosomes are going to digest the old and inactive organelles. We also are going to have lots of cytoskeleton within the neuron itself. These little branches on the top are going to be given their shape by the cytoskeleton. The axons, or the axon down here, is given its shape by the cytoskeleton. So this very unique shape of the neuron is maintained by a network of filaments, the cytoskeleton inside of the cell. And in particular, we have lots of microtubules making up that cytoskeleton. And why do we need so many microtubules? Other than just maintaining the shape, these microtubules are the highway system within the cell. And from the Nissl bodies, which are those very dark patches, those dark rough endoplasmic reticulums that I drew the arrows to up here, those Nissl bodies are going to produce chemical compounds, which then need to be transmitted and carried down to the axon terminal, the length of the axon. As we're looking at these extensions of the neurosoma, or the cell body of the neuron, we have dendrites. Dendrites tend to be the more numerous and shorter extension. I need to emphasize they receive the signal. They carry signals to the neurosoma. And then as the signals reach the neurosoma is going to process it. We can have an excitatory signal, an inhibitory signal. Different signals saying different things can converge, and the neurosoma is going to take all those little tiny pieces of information and make a decision. After the decision is made, if the neurosoma has reached the threshold voltage for sending a signal on and propagating the signal, that signal is going to be sent away from the neurosoma via the axon. And as we think of the axons carry signals away from the neurosoma. Usually, there will be multiple dendrites and a single axon, but occasionally there will be some variations. But as we look on the screen right now, this is your classic multipolar neuron that we have on the screen. Now, as we look at the axon itself can be kind of interesting. The axon is, again, going to carry signals away from the neurosoma. For the most part, the axon doesn't have many branches. Every once in a while, there will be a branch that you'll see on the axon. And when we find that branch, we call it an axon collateral. So here we have our axon. And then this branch coming off over here will be called an axon collateral. And these axon collaterals are unique. They're I should say unique and useful for carrying signals to very distant points of the body. So sometimes the neuron is going to need to propagate a signal to multiple points very rapidly, and it'll use axon collaterals to do that. Within the axon, there is some fluid referred to as the axoplasm, the cytoplasm of the axon. And the axoplasm is unique in that it's going to have a very tightly regulated ion concentration to help regulate the electrical activity of the axon. Just like we call the cell membrane of a muscle a sarcolemma, the cell membrane of an axon, or the plasma membrane of an axon, is referred to as the axolemma. It's worth emphasizing again that we'll have typically one axon per neuron. And this typically, every once in a while, there'll be an exception. I want you to think of axons being singular, one axon per neuron, and typically multiple dendrites per neuron. And again, there are some exceptions. And then finally, it's worth emphasizing that the axons may or may not be wrapped in an insulating protein called myelin. So on the screen right now, we can see myelin as the white protein that's in segments wrapped around the axon, and that myelin serves to regulate the speed that the electrical signal travels down the axon. Now, eventually, we get to the end of the axon, and we get to the axon terminal. Here is that myelin in more detail wrapped around the axon. And once we get to the very end, there's what's known as terminal arborization. An arborist is somebody who works with tree. Arbor is a root word that refers to tree. And when we think of arborization, I want you to think of at the branching out like tree branches or twigs at the end of a bough. So there's going to be many small, tiny little branches at the end of the axon terminal. And then at the very, very end, we have a swelling where it bulges outward. This is referred to as the axonal bulb, oftentimes. And this axonal bulb is going to contain vesicles filled with neurotransmitters. And then those neurotransmitters, when properly stimulated, or when those vesicles, when properly stimulated, will exocytize or secrete the neurotransmitters into this little teeny weeny tiny gap called the axonal cleft. And then those neurotransmitters are then going to initiate another action potential on the adjacent cell membrane from the next cell. And that's all we have for this recording. As always, if you have any questions, please feel free to post them on the class discussion board or to shoot me an email, and happy studies.